Let's move on to objective 2. We're going to speak about sort of the chemical background, the mechanism or the biochemistry behind this communication. So we're going to describe the chemical messengers and then the related receptors that are associated with the peripheral nervous system, okay? And again, really focusing on the autonomic division of the peripheral nervous system. So let's kind of backtrack and talk about the big picture here again. So we really want to make sure that we have the context before we start adding in these details. And so we're thinking about a preganglionic neuron synapsing onto a post or communicating to a postganglionic neuron. And so we know that at these synaptic communications, we know that there are certain players that we talked about that are involved. There has to be a neurotransmitter and there has to be a receptor to make that synaptic transmission occur. Okay. And so what we're going to look at now is the specifics of the neurotransmitters, excuse me, and the receptors that are involved in order to make the synaptic transmission between these autonomic neurons occur, right? And so we'll first start by looking at the receptors that are involved. There are cholinergic receptors, and cholinergic receptors are receptors that specifically bind acetylcholine, which is our most abundant neurotransmitter. And then adrenergic receptors are receptors that bind norepinephrine. Now, norepinephrine is sometimes a hormone. It can be a neurotransmitter or a hormone. And you may also hear this being referred to as adrenaline. So adrenaline and norepinephrine are the exact same, adrenaline and epinephrine are the same thing, and then norepinephrine and noradrenaline are the same thing. So if you're reading up in other texts or other sources, you may see that term, and it's the exact same neurotransmitter or hormone, okay? So these adrenergic receptors are specific to norepinephrine or epinephrine, which we'll see. We've also got some very specific neurons, okay, and then neurotransmitters. So preganglionic neurons, we talked about preganglionic neurons on the sympathetic side, preganglionic neurons on the parasympathetic side. All preganglionic neurons always secrete acetylcholine. Very first thing we want to make a note of, okay? Whether it's sympathetic, parasympathetic, all of them, once they are a preganglionic neuron, they release or secrete acetylcholine. In other words, if we look at the axon terminal of these preganglionic neurons, and if we look inside those synaptic vesicles, we will always see acetylcholine being stored there, okay? Parasympathetic neurons on the postganglionic side secrete acetylcholine as well, but then on the sympathetic side, they secrete norepinephrine, okay? So when you look at the postganglionic neurons, there's the split, there's that division. A postganglionic neuron on the parasympathetic side secretes acetylcholine as well. Therefore, both parasympathetic neurons secrete acetylcholine, if you're doing the math here, but on the postganglionic side, a sympathetic neuron will secrete norepinephrine or epinephrine, okay? And then as far as hormones of the adrenal medulla, remember we said that the preganglionic neuron could synapse onto our clomiphene cell, and our clomiphene cell would behave like a postganglionic neuron, and that clomiphene cell would secrete 80% epinephrine or 20% norepinephrine. Okay, now epinephrine and norepinephrine are somewhat interchangeable. They just have different affinities, but they have pretty much the exact same function, and they're just secreted in different proportions. Okay, and we'll talk about how that becomes relevant going forward. So the backstory, not the backstory, but the take-home message is both our parasympathetic neurons secrete acetylcholine, whereas our sympathetic neurons, only the preganglionic secretes acetylcholine, whereas the postganglionic secretes norepinephrine or epinephrine, and then the adrenal medulla clomiphene cell, which behaves like a sympathetic postganglionic neuron, is also secreting 80% epinephrine and 20% norepinephrine. Alrighty. So here is what that looks like in the sympathetic nervous system. Because we have 3 different routes of communication, and for each of those we have 2 neurons, so to speak, we want to be able to really identify what's happening here. So let's look at our first route of communication. So this is a ganglia that is a part of the sympathetic chain. So we have a preganglionic neuron. It's going to secrete acetylcholine. That makes it a cholinergic neuron. that acetylcholine will bind to a cholinergic receptor, right? Only acetylcholine can only bind to cholinergic receptors, but we have two types of cholinergic receptors, which I'll get into going forward. And then that postganglionic neuron, because we're in the sympathetic side, that is going to secrete a different neurotransmitter altogether. All right, it's going to secrete norepinephrine mostly. And norepinephrine, we said, binds to an adrenergic receptor, right? An entirely different type of receptor. Our second route of communication was a preganglionic neuron, also releasing acetylcholine. Remember, we said all preganglionic neurons release acetylcholine. So this is also a cholinergic neuron. That is going to synapse or bind onto a cholinergic receptor on our adrenal medulla chromaffin cells. So because these chromaffin cells within the adrenal medulla are acting like postganglionic neurons, they've also got the receptors, right? The receptors on the membrane in order to bind that acetylcholine. So they also have the presence of cholinergic receptors on their membrane. And when that acetylcholine binds to this cholinergic receptor, these cells will release epinephrine 80% of the time, norepinephrine 20% of the time, and that will bind to an adrenergic receptor as well, okay? Our third route of communication, a preganglionic neuron, also cholinergic, also releasing acetylcholine, binding onto a cholinergic receptor on the postganglionic neuron, right? This is how we relay that synaptic message or communication. And then that postganglionic neuron also releasing norepinephrine, which will bind to an adrenergic receptor on the effector tissue, okay? The only difference between this and the first one is that this ganglia is a part of the sympathetic chain. This ganglia is a collateral ganglia, so it's not a part of the sympathetic chain. And then looking at our effector tissues here, which can be heart, muscle, smooth muscle, gland, you name it, all of those tissues have these adrenergic receptors to bind this epinephrine or norepinephrine and to relay that communication, that signal, which will bring about sympathetic function, okay? Contrasting that to the much simpler parasympathetic nervous system. So in the parasympathetic nervous system, we have a longer preganglionic neuron, as we mentioned. That is also that is also cholinergic, so it's going to be releasing acetylcholine. Please bear in the back of your mind, all preganglionic neurons release acetylcholine, okay, and I'm repeating that very intentionally. And then that is going to bind to a cholinergic receptor, and then that second neuron will then release acetylcholine as well, because both of our parasympathetic neurons release acetylcholine. So there's no norepinephrine involved here. There is no epinephrine involved here. There are no adrenergic receptors involved here, okay? So the second neuron also releasing acetylcholine, but that acetylcholine is going to bind to another type of cholinergic receptor, alrighty? And so on that basis, we have two types of cholinergic receptors. We have a cholinergic receptor that is a nicotinic receptor that is located on the postganglionic neuron. So it's at the autonomic ganglia. And then our second type of cholinergic receptor, which also binds acetylcholine, is a muscarinic cholinergic receptor. And that is found on our effector tissue. And that is a different type of receptor. So nicotinic receptors are on our, going back to the sympathetic schematic here, they're on our postganglionic neuron. So they're here on this postganglionic neuron, they're here on our chromaffin cell, they're here on this other postganglionic neuron as well. And then they are here as a part of the parasympathetic postganglionic neuron also. Whereas muscarinic receptors are only found on the effector tissue, and they can relay the postganglionic signal to that effector tissue, okay? We'll talk about the other differences between these two types of cholinergic receptors, but this is something that we introduced before. Nicotinic receptors are fast receptors. Muscarinic receptors are slow receptors, okay? That's something you might want to make a note of, but nicotinic receptors, the receptors are also acting as channels. So when that acetylcholine binds, it automatically opens up a channel and we have that fast response. Muscarinic receptors are slow receptors. So what are they going to be coupled to? If a muscarinic receptor is a slow receptor or a metabolotropic receptor, what does it have to be coupled to?
A G protein.
A G protein, excellent, thank you. So that is correct. So a muscarinic receptor has a different mechanism altogether. It's going to take a couple steps and a couple second messenger, that entire pathway, that signal transduction pathway. And so the first thing is that it's going to have to be coupled to a G protein. Alrighty. Okay, so that's kind of just introducing that, and we'll sort of fill in some of those gaps as we go along here. So there are two types of cholinergic receptors here. We've got the nicotinic, which is an ionotropic receptor. The receptor is also acting as a channel. So as soon as that acetylcholine binds to that nicotinic receptor, it's going to open up that channel. We're going to see movement of ions, and that's going to immediately, depolarize the membrane and bring about that synaptic signal. Okay. Muscarinic receptors, on the other hand, which are only found on the effector tissue, okay? Nicotinics are found on the postganglionic neuron or the adrenal chromaffin cell. Muscarinics are found on the postganglionic tissue, effector tissue, and not postganglionic, so I think I said that wrong. So muscarinics are found on the effector tissue, right? So that's heart, muscle, gland, etc. And then the muscarinics we said are metabotropic. They are coupled to a G protein, and their effect is going to be different depending on what tissue they're located at. Okay, so very specific to the location of that target tissue. And I kind of want to explain again the reason why we see these two types of receptors in these two different locations. So if we go back here and we think about this fast response, when the signal comes in from that preganglionic neuron, it needs to be a rapid communication for that signal to be transmitted, and then the postganglionic neuron to also release its neurotransmitter. And so this is why we have that fast response here at the ganglia, right? That nicotinic receptor also acting as a channel to bring about that rapid, fast depolarization of the postganglionic neuron and really minimizing the time or the delay between the signal. The muscarinic receptor, it's a bit slower because the response here is a bit more tailored. It's a bit more customized. So if we're in the heart muscle, something different is going to have to happen. If we're looking at glandular tissue, it's a different response. If we're looking at the respiratory system, the lungs, it's an entirely different response. And so because of that customization, because of the individualization of the responses on the effector tissue, it's more of a sequential, sort of stepwise response that needs to happen. And so it's going to be a slow response as well. Okay. All right. Let's look at the locations here again. So I think we illustrated this on those images, but just to kind of tie it here together. So the autonomic postganglionic neurons have nicotinic cholinergic receptors, so those fast receptors. The effector tissue on the parasympathetic side has the muscarinic cholinergic receptors, so those slow receptors. And then if we think about other peripheral tissues or other branches of the peripheral nervous system, skeletal muscle also has nicotinic cholinergic receptors. And we'll talk about this in our next unit, but skeletal muscle is another one of those areas where there's not a customized response, but we do want for there to be a rapid response. Whenever I think to move my arm, my face, my limbs, I want that to be rapid. I want that to be nimble. And so in the skeletal muscle system, where we have voluntary control, we also see this fast nicotinic cholinergic receptor, okay? And then there's a really small exception, sweat glands. So sweat glands also have cholinergic receptors on the sympathetic side, okay? And the reasoning here is that as a part of the sympathetic response, we want to be able to sweat in order to lose heat. So whereas most of our glands, increased secretion is a function of parasympathetic, right? If you're increasing your digestive juices, increasing your tear production, that's going to be a parasympathetic function. But for a very small exception, the sweat glands, we do want increased secretions as a part of the sympathetic response instead, so that if you're running away from a threat, if you're anxious, nervous, a part of that sympathetic influence, we do want you to be able to lose heat, and that is done by producing more sweat, okay? All right, I'm gonna pause here before we get into these different types of receptors, just see what questions we have. All right. So we just talked all about the cholinergic receptors. And remember, cholinergic receptors are unique in that they bind acetylcholine only. There are two types, nicotinic and muscarinic. Here, let's look at the adrenergic receptors. So these are the ones that bind norepinephrine and epinephrine, okay? There are two main classes. They can be alpha receptors or beta receptors. And then within that, there are further subclasses. We can have alpha 1, alpha 2, we can have beta 1, beta 2, and beta 3. Now, these are all coupled to G proteins. So these are all metabolotropic receptors, right? There are no fast adrenergic receptors. Okay, I'll say that again. There are no fast adrenergic receptors. All adrenergic receptors are metabotropic receptors in that they are all slow in their response and they are all coupled to a G protein. Okay, so that's alpha 1, alpha 2, beta 1, 2, and 3. The difference between these types of receptors is their affinity. So whether or not they prefer epinephrine or norepinephrine, as well as their specific tissue location. Okay, and that's what we'll go on to kind of speak about here. So all of our alpha adrenergic receptors, that's kind of the first division, the ones that are alpha. These are mainly located in our effector organs in the sympathetic system. So heart, smooth muscle, you name it. They're mostly on vascular smooth muscle, actually. They're the most common throughout our system. They are usually excitatory. So that is, they're going to bring about an excitatory response. And if we think about the blood vessel where they're mainly found, this is going to be contraction or constriction of the blood vessel. So the type of response that they bring about is going to be excitation to the tissue, which is contraction in terms of the blood vessel. And then their affinity is usually greater for norepinephrine, right? They prefer norepinephrine. And so if there's an equal amount of norepinephrine and epinephrine around, they're going to grab onto that norepinephrine at higher proportions. And so that's the alpha receptors. That is going to govern both alpha 1 and alpha 2, which are mainly found on vascular smooth muscle. That is the smooth muscle that lines our blood vessels. Let's talk about the beta receptors. So beta receptors all activate cyclic AMP. So they have a second messenger, and we'll talk about this going forward, but their second messenger is going to be cyclic AMP. That's what's going to help bring about cellular response. And then they have different affinities, right? Whereas all of the alpha receptors prefer norepinephrine, beta receptors have different affinities among themselves. So beta 1 receptors are specifically located on cardiac muscle as well as the kidneys. They again are excitatory, so they're going to help to increase heart rate as well as increase kidney function. In other words, excite that tissue. They have equal affinity, so if there's equal proportions of norepinephrine and epinephrine around, they're going to bind them with equal affinity. They don't have a preference, essentially. Whereas beta 2 receptors are located in different locations. So they're on some blood vessels. They're usually also on the lungs. This is not mentioned here, but you can go ahead and put it on in your notes. So some blood vessels, smooth muscle of the respiratory airways or in the lungs. And then these are usually inhibitory to the tissue. In other words, they are not going to cause contraction or stimulation or excitation. They are going to cause inhibition or relaxation. And if you think about their location on the lungs, that will help you make that connection. If I'm running away from a bear or a tiger, I don't want to constrict my airway. I want to relax my airway so that I can bring more air in. And so the sympathetic influence to the lungs has to be an inhibitory effect to the tissue, okay? I'll say that again. Because of the strategic location of beta-2 receptors on the lungs and on some blood vessels, specifically blood vessels going to the heart or to our muscles. And if you think about the functional aspect of the sympathetic nervous system, right? Fighting, fleeing, freezing. If I'm running away from a threat, I want to open up blood vessels to bring more blood to my heart. And so I don't want to have an excitatory effect that's going to constrict those vessels. I want to have an inhibitory effect to send more blood to my heart, to my skeletal muscles to run away, okay? Same thing with the lungs. I don't want to excite my airway, excite those smooth muscle cells. I want to relax those cells so that I can open up the airway, bring more air in, and that is an inhibitory effect on the tissues. Alrighty. In terms of their affinity, they have a greater affinity for epinephrine. So kind of the opposite scenario here as what we saw with the alpha receptors. These receptors, beta-2, prefer epinephrine over norepinephrine. And then the last group here, which are beta-3 receptors, these are located on adipose tissue, so on our fat cells. They are also usually excitatory, and then we see equal affinity for norepinephrine and epinephrine. So beta-1 and beta-3 have equal affinities for epinephrine and norepinephrine. Beta-2 prefers epinephrine, whereas both of our alpha receptors prefer norepinephrine, okay? And so as we kind of wrap up looking at these receptors, we're going to get into the mechanisms of how they act probably next time, or we can kind of introduce that here. But I want you to think about making a table as a study tool for these receptors. So you're going to draw out a table. You're going to put all your receptors, alpha 1, alpha 2, beta 1, 2, 3. You're going to put their location, so put exactly what tissues they're found on. You're going to put whether they excite or inhibit their tissue, right? Whether they have an inhibitory effect, which is more likely relaxation of that muscle, or whether they have an excitatory effect, which is more likely constriction or contraction. You're also going to put their affinity, right? So what neurotransmitter do they prefer? Do they prefer epinephrine? Do they prefer norepinephrine? Or do they not have a preference? Is it equal affinity? And then the last column that you're going to put is basically going to be their second messenger. So we'll talk about the various mechanisms of each of these receptors, but they have different second messenger. So and they do a different thing to their second messenger. So some of them are going to activate their second messenger. Some of them are going to inhibit their second messenger. And we'll probably get into that next time we meet, but you're going to want to kind of create a table so you can fill in that detail and it will make a little bit more sense. Just, reviewing this and studying this quickly at a glance.